The overall goal of this procedure is to reliably transfect human THP1 macrophages with high cell viability and no cell activation. This is accomplished by first pre-differentiating THP1 monocytes for 48 hours. The second step of the procedure is to detach the premature THP1 macrophages. The third step is to transfect the cells with siRNA or plasmid DNA using the nucleofector technology of Lonza. The final steps are to reseed and further differentiate the cells. Ultimately, successful gene knockdown or overexpression can be shown through quantitative real-time PCR, Western blot, FACS, and so forth.
The main advantage of this technique compared to other transfection approaches such as lipofaction is that we can achieve high transfection efficiency together with high cell viability, and that we do not alter macrophage function and behavior.
Unfortunately, this technique is not suited for high throughput applications, as no more than 10 transfections can be performed at a time.
For this protocol, have cultured THP1 cells in rPMI 1640 with 10% FCS and with 1% pen, strep, L-glutamine. 24 hours before the transfection, split the cells and provide them fresh media. The next day, pre-differentiate the cells by seeding 10 to 15 million cells in a 75 square centimeter tissue flask with the following additions to the supplemented rPMI medium: 1% non-essential amino acids, 1% sodium pyruvate, 10 nanograms per milliliter PMA, and 50 micromolar beta-mercaptoethanol. After 48 hours, aspirate the media and replace it with 6 milliliters of Accutase 1, warmed to 37 degrees Celsius to detach the cells. Incubate them for 30 minutes. During the incubation, prepare sufficient transfection medium and cultivation medium for the post-nucleofection care of the cells. After 30 minutes, check that the cells are detached and look round. If some cells are still attached, gently pump the flask with a micropipette or use simple agitation to detach them. Do not use a scraper. Transfer the suspension to a 15 milliliter tube and centrifuge them for 5 minutes at 300 Gs and at room temperature. Remove the supernatant by aspiration and resuspend the cell pellet in 1 milliliter of rPMI medium pre-warmed to 37 degrees Celsius. Count the cells and make microfuge tube aliquots containing 2 to 2.5 million cells for immediate transfection.
A speedy execution of this protocol is essential to avoid loss of cell vitality. Be sure to have all materials prepared in advance.
First, centrifuge all the transfection aliquots for 10 minutes at 250 Gs and at room temperature. While they are spinning down, load all nucleofector cuvettes with half a microgram of plasmid DNA or 1 microgram of siRNA. Use highly concentrated dilutions so they occupy a minimal volume of the cuvette. Now, aspirate the supernatant from just one of the cell aliquots. Resuspend the cells to 100 microliters in nucleofector solution. Do not let the cells remain exposed to the solution for more than 15 minutes. Transfer the cells to the cuvette and mix them around with gentle tapping. Then, transfect the cells using program Y001 on a Model 2B nucleofector. Using a disposable pipette, move the electroporated cells to a reaction vial and immediately add half a milliliter of pre-warmed transfection medium to the cells. Now, keep repeating the process with each aliquot of cells, one by one, until they are all transfected. After completing the nucleofection, transfer each transfection of cells to a well of a 6-well plate with 2.5 milliliters of warm transfection media. Mix each well thoroughly with a micropipette. After letting all the loaded plates incubate for 4 hours, check the status of the cells under a microscope. After 4 hours, most of the cells should be adhered to the plate. If needed, at most another hour of incubation will provide sufficient cell adhesion. Working one well at a time, aspirate away the transfection medium from the adhered cells and replace it with an equal volume of warm cultivation medium. Be careful to avoid detaching the cells during this step. Now, incubate the cells for as long as needed. If necessary, replace the media every 48 hours. Plan to apply treatments so that they end when the transfected DNA or RNA is at its peak effect. When treating the cells with effectors, use a serum-free medium without PMA and without beta-mercaptoethanol. Be careful to not let the cells incubate for more than 24 hours without serum. The protocol was used to transfect THP1 macrophages with siRNA. Cellular morphology was assessed according to flow cytometric measurement. The rate of apoptosis and necrosis in the cells was low in both transfected and control cultures. However, careful counts revealed that both apoptosis and necrosis were slightly higher for the transfected samples. This could be because transfected THP1 macrophages were more difficult to detach from plates than untransfected cells. Overall, transfection rates were above 90%, as analyzed by flow cytometry using fluorescent siRNA. Transfection efficiency was also confirmed by fluorescence microscopy. The functionality of the transfection was shown by genetic knockdown of IL-10Rb expression using short interfering RNA.
Once mastered, the transfections can be performed in 1 to 1.5 hours if it is performed properly.
While attempting this procedure, it is important to remember that the culture medium has considerable influence on the performance. This is outlined in the text protocol.